propagation of plants, now that they have done it for a long enough stretch of time, it's really becoming a reliable tool in order to mass produce plants. And so I don't know if you have ever planted potatoes, but just planting the tuber, if you see it sprouting like that, you can just plant that in the ground and it will start growing. And so they actually sell what are called seed potatoes, which the first time I heard that, I'm like, what? That confused me for a while until my postdoc mentor said, oh, no, what they do is they sell potatoes that they know are disease-free, but they're tubers. And as long as they have some nodes on them where new branches and leaves will start growing, you can plant that in the ground. In fact, you can chop up a potato. And as long as each section has a node on it that will allow for sprouting, you can do that. So that's what we used to do. We'd order these seed potatoes and then we chop them up so we get more bang for our buck and plant them. If you're curious, that's a potato field. So with propagation, this idea that you can chop up plants and get them to grow into a full intact plant and thrive and grow indefinitely as long as you give it the right kind of care is pretty amazing. This quote is taken from Dr. Mancuso's book. I have it in the notes. The book is called The Revolutionary Genius of Plants. It's a really interesting book. And I actually talk about some of the ideas presented in there in my advanced botany class that will be offered this fall. And so if you have any interest in that, it's maybe not during the semester, but maybe just summer read. He does a very good job of keeping things non-jargon. So it's really easy, but I love this quote. Plants distribute over their entire body the functions that animals concentrate in specific organs. Decentralization is the key. We have discovered that plants breathe with their whole body, see with their whole body, feel with their whole body, and evaluate with their whole body. Spreading each function over the entire organism as much as possible is the only way to survive predation and damage. And plants can do it so well that they can even withstand removal of much of their body without losing functionality. And that is so true. I mean, think about it. If our heart stops, we're done. If our brain stops, we're done. But plants don't have that, right? They distribute all of that through their body. You can chop them up into tiny pieces and they'll regrow into an intact plant. So it really is an amazing process. And when you think about what has to happen at the cellular level, once they get pruned back or cut, it's really quite amazing. So we were looking at this briefly yesterday or Wednesday, and this idea that, especially if you make sure that there are meristematic regions of the cutting that you take, then you have a good shot of having a healthy stem cutting start to grow vigorously and produce what you need for the intact plant. And then, of course, getting the adventitious roots to form is another amazing process. So for this, make sure that you're up on the terminology, with the terminal or apical meristem region, the axillary meristem or lateral meristem, the node region. So make sure that you're getting that. I would like you to know the idea of the petiole, which is the leaf stalk, the actual, what would say the leaf, that's the leaf blade technically. So when you think about those stem cuttings, you can actually, if it's a long plant, you can take several stem cuttings off of that one stem. And you just, ideally, you want each one to have that, axillary bud. And then, did you use the root tone at all? Did you opt into that? So that's a nice little boost. If you dip the cutting into root tone and then put the cutting into a beaker or bottle of water, and then it's really fun to watch the adventitious roots form. So all those cells in that area are sensing that they've been wounded, and basically that gene expression really shifts greatly because those are actual stem cells that that form the stem tissue. And now they're like, oh, but now I don't have roots. When you take that cutting, they're like, I don't have roots anymore. I better grow those. And so they have to stop being stem cells, turn into root cells and start growing. That's a phenomenal shift in gene expression that's going on. It's quite amazing. And so for the plant to be that adept at figuring it out and then switching that massive gene expression pattern is pretty astounding. And so, you know, people do it all the time, and it's kind of, a lot of people know that they can do this, but when you get down at what's happening at the cell level, it's pretty amazing. And then I think I talked a little bit about this too. You can even take just leaves and get them partially buried in the soil. And you can start to see the little baby plants that's starting to grow in that area that was submerged into the soil. And so again, this idea of, you know, it's just a leaf. And so now those leaf cells have to figure out, I've been removed from the mother plant. I have to regrow an entire body. And so they will do that if you give them the right conditions. You can even do it with succulents. I don't know how much of that was provided at all, but just even taking a leaf off of a succulent and partially burying it like that into the soil, you can get little baby plants growing from that. So I think this is African violet leaves that you see here, where you can take like the leaf blade and the petiole and bury the petiole into the soil. And then I think I showed you this too, where they just laid the leaf. This looks like maybe a streptococcus, streptocarpus leaf, which is actually related to African violet. They just laid it flat on the soil surface and then made some wounds across the central vein and just gave it the right conditions and it started to grow. I don't know if there were any of this snake plant. Was that an option? That's always fun. Hopefully Duncan explained to you, you have to get it in the right way. Otherwise, you know, if you don't have it in the right orientation, the roots will actually form on the above ground portion. So that's a fun one to do. You can, you know, take multiple sections from the same leaf and plant them. And this one, if you did choose to do snake plant, just be patient. It does take quite a while for the roots to form, and then you'll slowly start to see new leaves coming out of the soil. I have some I used to have some in my house that I took from a cutting from the greenhouse, and it took a while to get the big new plant. But once you have it, snake plants are very used to dim conditions, shade tolerant. And so unfortunately, my living room is pretty shady. So they were amazingly happy in that area. So it's a great plant. They're very hard to kill. We didn't, I don't think, try any of this, but these are options. If you have the idea of just burying the tip, it's really kind of funky. You do that and just make sure it stays moist. And because it's buried into the soil, it'll start to, oh, okay, I guess I'll start forming roots and then a new shoot. And you can then make a cut and take that as a new plant. And so that's the fancy tip layering technique. And then you can do what they call the simple layering, where you just bury part of the stem instead of the tip. And they have it kind of anchored in there with a stake. And then you can see that slowly it'll start to form the roots, and then you can cut that off the mother plant.
Do the plants that send out like thunderbirds, are they just doing tip layering?
Pretty much, yeah. It's pretty fun to see that. We've just kind of, someone observed that and said, well, we probably can make a lot of other plants do that too, and they're right. Here's a good one, mound layering, where you mound up the soil above the branches. And now you buried them. So they're like, oh, I need to form some roots. And then you can get the root formation, and then you can cut that from the mother plant. So that's pretty cool. And then air layering, I've tried it once, it worked. I was amazed that I got it to work on the first one. You kind of damage or kind of strip off a layer on the branch. And then you cover it like with sphagnum moss typically to nice and moist, and then you wrap it so it's light, you know, cover the lights so it's dark in there. and it will start forming roots, and then you can cut it off the mother plant. So lots of options there to propagate. So keep those in mind as you have plants in your life. These are fun ways to, especially if you like that plant and want to create more of them, it's pretty easy. So this is just a short little introduction to propagation. And now the goal is in lab to keep that propagation that you've done going. And then hopefully you'll be able to plant it and take it home at the end of the semester if you'd like to keep it. Okay, so I'm going to talk a little bit more. The chapter where this is covered also covers plant biotechnology, I believe. And I'm going to save that, shift it to, I think, right after spring break, because I need to keep on track with what we're doing in lab. And so I want to make sure that we're covering root stems and leaves when you're doing root stems and leaves in lab. So hang on to the biotechnology part. We'll get to it. I'm not right now. Okay. Okay, so actually the next slide set we're going to get into is the photosynthesis idea. Okay. So as long as we're here, we've been talking about cells and tissues, the anatomy of the plant, just basic anatomy, and then we got into plant growth. But now I'd like to go a little deeper into how is the plant growing? How is it making its sugars and energy in order to stay alive? And so in this class, what I tend to do is so there's two parts to photosynthesis. There's so-called light reactions, and then the biochemical reactions that lead to sugar production. In this class, I'll talk a little bit about the light reactions, but we're not going to go into it in detail. We go into it in detail in plant phys. That's where you really get the deep training on that. This class, you get an intro, but I'm going to go into a little bit more detail on how they make their sugars, because I think it's important in a basic botany class. We're going to find out that plants do that differently depending on where they live. And so they've adapted different strategies. That's why you find, the cacti or cactuses, what's the plural of cactus? I always forget. Is it cacti? Cactuses? Maybe both are allowed these days. I don't know. Anyways, forgive me if I'm saying it wrong, but that's why they're in the desert and not here in the Midwest, well adapted to this area, and why certain plants grow only in the tropics, but not this far north. And so I think once you get a feel for that idea, it really makes it more pleasurable when you are traveling and seeing these different plants and learning about the different geographic regions. You can start to get a feel for why they're so successful in that area and perhaps not so much in other areas. Okay, so basically, for photosynthesis, one of the goals is to get ATP. And as you know from basic biology, adenosine triphosphate, that's a big thing for us too. That's what we like. Our cells and tissues work off of ATP for their energy. Well, plants are amazing. They can just make it through sunlight. And so wouldn't that be nice to go to the beach, lay out for like 15, 20 minutes, get some sun, and you're good? You're good to go for the day. You're all energized. You feel full. But just think how much less time would be spent cooking meals. I'm not a huge fan of, it just takes too much time to cook meals. So anyways, I'd love to just lay out and get all it done in 15 minutes for the whole day. But anyways, okay, so plants make their ATP using light. We're going to see how that works. Now we're zooming in back to the chloroplasts, that organelle. And so I'm going to briefly go over a little bit about the chlorophyll structure and point out a few things to refresh your memory. But the chlorophyll is green when the chlorophyll is green, the chloroplasts are green because they're chock full. The chloroplasts are chock full of that chlorophyll pigment. And so just like we were talking about the phytochromes absorbing different wavelengths of light, the red or the far red, the chlorophyll is designed and adapted to absorb green or red and blue light. And it reflects green. that's why it looks green to our eyes. It's reflecting and bouncing that off. It's like, I don't want green, get out of here. And so our eyes pick that up and we see that it looks green. Okay, so the chlorophyll pigment is super good at absorbing certain qualities of light. So you often see the photosynthetic reaction pathway written like this. 6 CO2 plus 12 water plus light. I usually put, when I'm writing this out above the arrow, I'll put CHL for the chlorophyll. You gotta have chlorophyll. Don't forget about the chlorophyll. And then once you get those reacting, you end up with C6H12O6 plus 6 oxygen plus 6 water. So I like to call this the big picture equation of photosynthesis. This is actually taking into account both the light reactions and part 2 of photosynthesis, which is the biochemical reactions for sugar. Okay, so this is like the grand idea. When I say C6H12O6, what sugar do you think about? Glucose. Ah, yes, absolutely. I do too. So I'm not a fan of this equation at the college level, even though I know why it's written this way, and it's not inaccurate. It's just that when I see C6H12O6, I just kind of go to glucose. But I just want you to know that glucose really is not the sugar that's being produced. That's not what they're making. There's a different sugar that they make, and that sugar gets sent through their whole body to other parts of the body that need that sugar. But it's not glucose. So it's a little bit confusing. Okay. This is also the big picture idea. All right. This is combining both what's happening in the light reactions and the biochemical that make the sugars. So you see here the photochemical reactions. They're trying to show the internal membrane here. They're showing just a little bit of it. And they're saying you got to have light. So that squiggly, anytime you see a squiggle light or a squiggle arrow, that usually means energy. In this case, it's sunlight striking the chlorophyll in that thylakoid membrane inside the chloroplast. So the chlorophyll absorbs that light. And what that does is it creates a situation where it kicks off an electron transport chain. I have this in words in a later slide, just it's okay. And so that's really important, and I'll get into that. And so another thing that it allows to happen is it strips electrons off of water. That's phenomenally hard to do. Water doesn't want to give up electrons. It's very partial to its electrons. You have to really zip it with some energy, zap it with some energy in order to get electrons. And so the sunlight energy and the way the plant has these special photosystems, it allows them to extract electrons from water and help send these electrons down this transport chain. So if you take electrons off water, one of the products is oxygen, which with regard to photosynthesis, oxygen is just a waste product. Oxygen doesn't fit into this anywhere. But certainly we are very thankful that plants are doing this because that's the oxygen we're sucking in and breathing. So for us, this is great. But for the plant, as it's doing photosynthesis, it's like, yeah, I don't really care about oxygen per se. In order to do photosynthesis, that's not what I need. I need electrons from water. And yeah, okay, one of the products is oxygen. I'll just kind of get rid of that and then we breathe it in. Okay. All right, so they're showing the light reactions, and then what they're trying to show is that there's 2 key products that are being made during the light reactions. You have ATP, which when they put it like that, so they're trying to say that's your energy, but NADPH is also there. That's also very important. So NADPH is a molecule, we call it a reducing molecule or a reducing agent. And it's also very good because we're going to see that both of these actually, ATP and NADPH, will be used as a source of electrons and energy when they help out the Calvin cycle. So the Calvin cycle is a cycle. It zips around and around and around. And this is part two. This is the biochemical reactions where you get the sugars getting produced. So this is where you see the carbon dioxide being put to use. So the plant will open its stomata during the day, take in carbon dioxide and feed that into the Calvin cycle. And as that's happening, it will go round and round and round. And after enough turns of the cycle, it has enough carbon in order to make its sugars. And the sugars that it actually is going for is sucrose. That's what it's actually going for. It's sucrose. In order to run the Calvin cycle, though, you have to have some energy input. You have to have ATP pushing certain key aspects of the reaction pathway. And NADPH has to be there to donate off some electrons. Okay. All right, so this is like the big picture idea. Here's another big picture idea. This is from actually the Plant Phys textbook. So this is the equation I prefer to teach off of, where instead of putting C6H12O6, basically you've taken all the numbers out of the stoichiometry of it, essentially, and you've stripped it down to the bare bones. And you just say, if you take a CO2 and you take a water and you have light and chlorophyll, you'll end up with CH2O. And if you see that written like that, that's the basic building block of a carbohydrate, okay? So that's the simplified version of C6H12O6. That's the bare bones version of it, CH2O. So when you take chemistry classes, they often will write it that way, and they mean that that's the basic building block of a carbohydrate. And then you see the oxygen that we have the ability to use. Yes.
You want us to know both equations or?
I prefer you know that one. This one's more appropriate. So the Calvin cycle is also called carbon fixation. If you ever hear that, they'll say that for like something fixation. They usually mean you're taking whatever it is, carbon in this case, and making something more complex out of it. So you're taking a CO2 and making a sugar out of it. And so that's usually what they mean when they say fixation of some chemical. It's called the Calvin cycle, named after Dr. Kelvin, or they call it the C3 cycle. They also call it the photosynthetic carbon reduction cycle, which the acronym is PCR. What do you think when I say PCR? What do you think? So I never use this. It's too confusing. Back before the PCR polymerase chain, they used to say that a lot, but I tend to say Calvin or C3 cycle. I tend to use that when I'm teaching. Okay, but just be aware there's a lot of names for the Calvin cycle. So this is another big picture idea where they're linking both the light reactions and then carbon or biochemical Calvin cycle. All right, so this is saying basically the same thing as the last one. If this works for your brain better, go ahead.
I have a question. So the Kelvin cycle, is it also like the Krebs cycle on the other side, or is that completely different?
No, the Krebs cycle is also a way to make some ATP, but the Krebs cycle is actually breaking down sugars. This is building up sugars. So it's kind of the opposite, like it's kind of opposite in intent, where this is building sugars and the Krebs cycle is breaking down sugars. So yeah, yep. Okay, so let's refresh the lovely chloroplast. You've had this, but remember, it's got an outer membrane. It has an inner membrane. And then it has an internal membrane that's floating around. And that whole membrane is called the thylakoid. Okay. Now we also give different terms to the stacked region versus the relatively unstacked, but for this class, let's just say that's the thylakoid. And then the watery stuff, if you crack open the chloroplast and peer inside and you see what is the thylakoid floating around in, all that watery stuff is called the stroma. That portion of the chloroplast is called the stroma. And then if you do some surgery and cut into the thylakoid membrane and look inside, it's empty in there. It's like a hollow, empty membrane. That's what they're doing over here. You call that internal portion the lumen. Okay, the lumen. So I do need you to know for sure, everything that's on this slide, all the terminology.
So the thylakoid is also.
Yes. Yep. So if I were to ask, what's the internal membrane of the chloroplast? It's the thylakoid. That would be how you would answer. So we have a lumen, our intestines, you know, if you peer into your intestines, that inner portion is called a lumen, basically. So it's not unique to a chloroplast. It's just whenever you look inside a membrane bound in compartment, which I guess your intestines are more like a tissue bound compartment. Okay. So let's look at the light reactions. So in the thylakoid, what we have happening is water will get split or the electrons get stripped from it. And again, this is really hard. You have to put in a massive amount of energy, which is sunlight energy coming in and the absorption of that by the special chloroplasts. Okay, so you need a source of electrons and you need some electrons zipping along this electron transport chain. So water is the ultimate source. You got to keep stripping electrons off of water. As you do that, you get actually two products. You get that oxygen, which I told you about already, but you also get this very useful proton. So really, when you think about the light reactions, I'm not really going to talk anymore about oxygen because that's really irrelevant, but I certainly am going to talk about protons and electrons. Those are super important. So inside the thylakoid membrane, in the lumen, this is happening. You get water, it's in that lumen portion, and water is getting stripped of electrons and protons. So you have protons starting to build up in the lumen. So hang on to that thought. Electrons are zipping along on an electron transport chain. And I'll show you a little bit about that. So electrons, they get passed along. I like to think of it as a game. You know, you have this electron getting captured by one of the players, and then they donate it off to the next one, who then donates off to the next one. It's like this big assembly line of passing the electrons or passing the hot potato from one to the next until it reaches the end molecule. Okay. As the electrons are moving along, it allows, there's a little bit of energy through that process that will ultimately allow for ATP to be made. Whoops, in the stroma. Looks like I didn't get the A to turn blue. So stroma. Does that show up well enough where you're sitting, the blue on the green? Is that okay to your eyes? All right, so ATP is made in the stroma, and the plant needs that ATP because we're going to find that the ATP is going to get used right there in the stroma for part 2. You get the sugars getting made. It's the Calvin cycle. The other thing that's happening in the stroma is your NADPH is getting created. So the very last player in the electron transport chain process is this molecule, NADP+. Don't forget the plus. So the NADP+ is the last one, like I'm waiting for my electron. I'm the last one. And then once it gets that electron, what happens is it reduces to this NADPH. And we call the, well, I think I mentioned it, the NADPH is called a reducing agent. So now just kind of think of like NADPH as an electron carrier. It's a molecule that's like, okay, I got this electron. Now I can go and share that with another reaction pathway going on. Right? So it's very useful if you get this made along with the ATP. So both the ATP and the NADPH are necessary for part two, the Calvin cycle, making the sugars. Okay, let me show you a picture of what all this is in words. Let me show you a picture now of that. So this is actually taken from the plant phys class. And it's kind of a simplified model of photosynthesis. So I teach off of this in plant phys, but we also get more detailed. We're not going to get into the details of what I do in plant phys, but there's a few key things on here I want you to be aware of. So the membrane that they have drawn here, okay, this membrane, that's the thylakoid membrane. Okay, so they've taken a chunk of the thylakoid membrane. And all of these big things that are embedded in, and then this PQ and the PC and these are all the players in the electron transport chain process. Well, not all. There's some stuff missing, but for this class, this is the basic electron transport chain. So do you see where water is? Yep, it's at the beginning and it's in the lumen area. Okay, so this is the internal part of the thylakoid membrane. If you peer inside the hollow portion inside of it. So water's in there. And as light strikes so PS1 and PS2, the PS stands for photosystem. Photosystem 1 and 2. They're just named after the order. So they discovered photosystem 1 first, so they named it 1. And then they said, oh, there's another one. Let's name that 2. All right. So those are very special portions of the electron transport chain. There's a lot going on in here, which I won't go into detail. But basically, photosystem 2 is very special because it has the ability to extract electrons from water. Photosystem 1 doesn't do that. Okay, so photosystem 2 is really special that way. It has the ability to take that sunlight energy and create a situation where it can strip electrons out of water. And so then you see this dotted line. They're saying, okay, water got stripped of electrons. And then the dotted line is coming through this photosystem. There's some other players here. And then what they're trying to show is that the electrons get passed from player to player, donor to recipient in this chain process until finally you see NADP plus getting that electron and getting reduced to NADPH. Where is that happening? What part of the chloroplast? Stroma, all right. So that's the watery portion that the thylakoid's floating around in. And that's really good to have NADPH there. That's a very convenient thing. Okay. So for this class, I'd like you to know, for the light reactions, that there are 2 photosystems, photosystem 1 and photosystem 2. Photosystem 2 is really special because it can actually strip electrons off of water. And then I don't really want details of who else is, as long as you understand that there's players in this game that they pass the electron down the chain, okay? And ultimately the electron gets to NADP plus. Okay, so understanding this electron transport chain process with many players in the game here. And the ultimate acceptor of that electron is NADP plus. Okay. Now, another thing going on here that I want you to key in on is that when water is getting stripped of electrons, they're showing that protons are one of the products. H plus. I don't care about oxygen for this. I mean, I'm thankful they produce oxygen, but I don't care about it right now. So you see protons are showing up in the lumen from that action. And then you see here too, there's some protons showing up. So again, there's a lot to this. And we go into it in plant phys, but basically what's happening here is as the electrons are flowing through this chain process, it creates a situation that allows for protons, we often use the word pump. Protons get pumped across the membrane. And I go into it more in Plant Phys of the actual, like, how does that happen? But it's quite an amazing process where this electron flow provides a little bit of energy in that process that can allow for this proton to come across the membrane, and, so to speak, pumped across the membrane and end up in the lumen. So this is also happening in you right now, in the sense that your mitochondria are doing this. The mitochondria are running an electron transport chain process, which allows for proton pumping across the membrane of the mitochondria. Why should we care about that? Well, if you have this happening over and over and over again, this electron flow over and over and over, that means you're going to get a lot of protons building up in the lumen. And now you have a proton gradient. Okay, a proton gradient. Anytime in science classes, if someone says, oh yeah, that creates a proton gradient, you should go, ooh, I bet somewhere, maybe they're making ATP. That could be one thing that they're doing. So if you get a proton gradient, more protons, okay, let me be more specific, a proton gradient across a membrane. Okay, so here's your membrane. For you, it's your mitochondrial internal membrane. In the chloroplast, it's a thylakoid. You get more protons in the lumen, less protons in the stroma. So you have a proton gradient across this membrane. By the laws of diffusion and all of that, these protons, they want to diffuse and even out on either side of the membrane. Because they're charged, they can't go through just the normal portion of the membrane. It won't work because they're charged. So they have to find another route. And so they go through the ATP synthase. And as they do that, it's amazing. They call this a little molecular motor. It allows this flow, actually creates a torque force that causes these subunits to rotate. And as they're rotating, it allows the ADP and the inorganic phosphate to react with each other. And then you get that lovely ATP. It's quite amazing that a flow of protons can cause a torque force, which then allows for the production of ATP. Where's ATP showing up? Stroma. That's also very good, because we're going to find out that the stroma, that's where you have the Calvin cycle or the C3 cycle cooking along. So now the ATP and the NADPH get made right there, and they're getting used for the Calvin cycle right there. It's very convenient. Questions on this?
Like with the building of the protons in the lumen, so like once there is a buildup of the
Protons?
protons in the lumen, then it just like causes a reaction to shift up through the
Yeah.
ATP synthase.
Yeah, because if you have this imbalance, we have more protons across the membrane, lots more here, not very much over here. By the laws of basic chemistry, they want to diffuse across. It's just like diffusion. It's like, you know, like if you open up a perfume bottle in here, eventually the perfume molecules are going to reach the far wall through the laws of diffusion. And so they want to diffuse across because their concentration is different. Did you have your hand up? Yeah.
So is the proton buildup a result of photosystem 2 stripping the electrons?
Yes. So this is happening over and over and over again, as all day long, hopefully, for the plant. They will continue to strip electrons off of water that's in the lumen. They just keep doing that all day long and sending those electrons through the electron transport chain. So that's one huge way that they get build up protons. And then this proton pumping, you see protons actually getting taken from the stroma and getting dumped into the lumen. And that's because of the dynamics, the energy dynamics of how the electrons flow, which you don't need to know about in this class. But basically it creates a situation where it allows the protons to get moved from the stroma to the lumen. And so the goal really, anytime you see an electron transport chain, whether it's your mitochondria, in the chloroplast, or when I teach Gen Micro, it's in their cell membrane. They create situations that allow for an electron transport chain process. And I can't think of an exception. It creates a proton gradient. It's like you can't divorce the two. You get an electron transport chain, you have a proton gradient getting established. No matter if it's a mitochondria or chloroplast. And that's how you're making your ATP right now. This is also going on with different players in the game. Mitochondria do different players, but it's the same basic process. Get your electron flow going. That will create a situation for a proton gradient, which will allow for ATP. So it's kind of a universal strategy. It's really quite amazing. It's a very old, microbes did this first. To be honest, it's microbes that came up with this billions of years ago. Okay, other questions on this? All right, so hopefully during the day, the plants will open their stomata, start taking in CO2, they'll start running this reaction. And so they'll start generating some nice ATP, they start to get their NADPH, and then if their stomata are open, if they've been well watered, they're not in any drought condition, they will be taking in their CO2. And that's where we see things cooking along in the stroma area, the watery portion of the chloroplast. You'll see the carbon fixation, C3 cycle, Calvin cycle. So this is actually named after Dr. Calvin. His full name is Dr. Melvin Calvin. I think he might have been teased a little bit when he was younger. He actually went to Michigan Tech University. He did his bachelor's degree there. So when I was there, the biochemistry professor, I took advanced plant biochemistry, and he went on and on about Dr. Calvin, rightly so. I mean, he's really famous in the plant world. You probably are like, who? But he's famous. And he's the one that was credited with figuring out the final problems they were having with the Calvin cycle. You have to realize this was quite a while ago. Okay, so the technology, not the greatest. And they kept getting stuck on what does the CO2 react with? They couldn't figure it out. And they tried all these experiments. Nothing was working. He came into the research and said, well, they at least figured out that the start of the cycle, there's a 3-carbon sugar. They had that figured out and isolated. So they're like, okay, we know what this 3-carbon sugar is. We know that carbon dioxide has 1 carbon. So to get a 3-carbon off of that, you must need a 2-carbon molecule to react with the carbon dioxide. So you get to 3, right? 2 + 1 is 3. That's logical. But they couldn't find it. They couldn't find this 2-carbon molecule. So then Dr. Calvin came along and said, well, there's more than one way to get to 3. What if you take 6 and divide it by 2? That's 3. And that blew it open. They're like, oh. And so they realized it was a 5-carbon sugar that was reacting with the carbon dioxide that came in. And what happened, they finally, once the technology got a little bit better, they realized there's a very unstable 6-carbon sugar that was produced. And then it quickly breaks down into 2 3-carbons. And so they kept missing that. They didn't see that happen without the technology. And so that sugar is called ribulose 1,5-bisphosphate, which luckily we abbreviate down to RUBP. And that's how I'll talk about it from this point forward, because I really don't like saying that big word. Okay, so the 5-carbon sugar is RUBP. That's what's reacting with the CO2. It forms 6 carbons, and then it breaks into 2, 3 identical molecules of this certain sugar. So that's what we're gonna talk about on Monday. We're gonna get into the Kelvin cycle in a little bit more detail, okay? But we'll stop there. Have a good weekend. At least this time it's not freezing.